So our speaker today, as Samantha said, is Dr. Elise Zipkin. She's a Red Cedar Distinguished Professor in the Department of Integrative Biology at Michigan State University, and also the Program Director for Ecology, Evolution, and Behavior. So prior to joining Michigan State, Dr. Zipkin did her undergrad at the University of Michigan in mathematics and applied ecology, a master's at Cornell University in natural resources, and she got her PhD from the University of Maryland. And then Dr. Zipkin also worked at the USGS as a quantitative ecologist. Dr. Zipkin has built a research program that uses mathematics to understand ecological communities with a focus on biodiversity loss and charting pathways to reverse it. And she regularly works with management agencies to translate the results of her research for conservation. Dr. Zipkin's honors include being named a Fulbright U.S. Senior Scholar, an International Ecology Institute IPRE Laureate, and an Ecological Society of America Early Career Fellow. And so I'm excited to introduce Dr. Zipkin today to talk about making the most of available data, how integrated modeling can expand the usability of individual data resources. So take it away.
Thank you so much for the introduction, Eric and Samantha, for inviting me. I'm going to try and share my slides here. So let's see if that worked. Can anybody tell me? Because I don't know. Just like a hands up or yes.
That looks great, Elise.
Okay,
Perfect.
great. Thank you. Okay, great. So I'm going to talk today a little bit about some of my lab's work on data integration. And I titled this talk, Making the Most of Available Data, because, a lot of us are familiar. There's this divulge of lots of different kinds of available data types now on many different disparate things. And the reality is, we might be able to get a lot more out of that if we put them together. And I think Neon is a perfect example for this. I'm not going to be talking, I will mention some Neon data that we're using. I'm not talking specifically about it, but when Neon first came online, when I was first hearing about it, 10 years ago or so, I was thinking, wow, this is going to be an amazing resource in 30 years. But what can we think about, how can we think about using these data in a shorter time frame, more on the, faster time scales? Like, what about them is going to be so special? And I think, obviously the kind of standardized protocol, the large spatial area that the data cover are amazing. But I think one of the really awesome things that they are going to provide, or they are providing, at least for my work, and I think several others, is that we can integrate them with data sets that already exist and get a lot more out of what we already have, considering the unique placement of the Neon data sites. Okay, so that's kind of the motivation for this. So, all right, let's see. Okay, can I change a slide? Yes, all right, did that work? So most of my research is thinking about addressing the biodiversity crisis, right? So all of these are pictures actually that were taken, not by me, but throughout the state of Michigan where I'm located. And, we have this amazing swath of biodiversity here locally, of course, in the country and actually internationally. But all of us know we're losing species at really high rates. And so thinking about how we reverse these trends and preserve what we still have left, I think does require really revolutionary approaches. And I think that, as I think about this, that's something both in terms of data, so kind of new, big, large scale data that are going to help us solve this problem, and also innovative statistical modeling approaches, which is what my lab works on. So when I was in graduate school, I took a class from Stephen and he put up this picture and said, we are in the golden age, he said, of theoretical ecology. So this, in case you don't know, is a picture from 1927 on a really famous physics conference that was kind of at this edge of where, physics really kind of took off in at least the last century. And so there's all these famous people who are there discussing ideas. And, as I've come to learn more about ecology and statistics and statistical ecology in particular, I think we're really at this golden age of statistical ecology, actually. There's a lot of people who are really interested in these kinds of approaches and in developing approaches. We're in this time where we have these big data that are going to be able to allow us to answer some of the questions that we previously couldn't answer. And we have this computational power. So I think that this is a very exciting time to be thinking about statistical ecology and big data questions and macro scale questions. Okay, so today I'm mostly going to talk about this framework that my lab has been developing called integrated community models. And the basic idea with integrated community models is that we're putting together multiple different data sources, so that's the integrated part, on multiple species within a community. And so there's been other work where people are thinking about how do we model, more than one species. So instead of a species by species approach, how can we answer questions related to whole communities? And there's also been some approaches where we're thinking about let's aggregate the data. So my lab has kind of been moving forward on this line of research to think about general approaches for that. So integrated community modeling is comprised of two basically separate frameworks for thinking about statistical models. The first is hierarchical community models, and the second is single species integrated models. And so to really understand what we mean by integrated community models, I want to first go through each of these separately and show you some of the benefits, what they are and why we're doing them. So the first is hierarchical community models. And so I'm going to go through this the same way with single species models, but I want to talk about first, what are hierarchical community models? So the basic idea with hierarchical community models is that lots of survey types produce data on multiple species, even if that's not necessarily something you want. So for example, thinking about going to collect a point count survey in the middle of the woods, or walking a transect and turning over stones to find salamanders. You're naturally going to be able to see with those survey types multiple different species, right? So there's just this natural way where we can be collecting data for multiple species simultaneously. Now, in the past, those analyses for those species would be conducted each one species by species. But the idea with the hierarchical models, we want to put all those data together into a single model. So from a single data source where we're collecting data, putting together all the data on a community. So the basic idea is that we're modeling some biological process. For simplicity, we can think of it as something as the occurrence of a species or the abundance of, individuals in a species, that sort of thing. And we have some factors that affect that. Maybe those are some environmental covariates. But unfortunately, we can't get at the true biological process exactly. Instead, what we do is we collect data on that process. And those data, which might be something like species counts or presence absence or all other sorts of things, they give us information about that true biological process. But what gets us from our data to our true biological process is that sampling process and factors that may be influencing our ability to sample the species effectively. So we call those detectability. And really the idea is that our survey data are imperfect. So they're not showing us exactly what's going on with the biological process, and we have to think about that. So now I'm just describing how we would model this for an individual species, but then the next is we want to take this true biological process for an individual species, the data that we collect on that species, and put that together into a model and link those at the community level. So what do I mean by that? Well, for example, let's say we were modeling species occurrence, like an occupancy type model. What we would want to assume then is that each of the covariates in that occupancy model and that detection process all come from a common community level distribution, so that there's a mean among the species and a standard deviation across this. So what does that mean exactly? It means, like, let's say I was trying to understand what the effect of, forest cover was on a forest interior bird species. So I would say that that effect, instead of estimating it separately for each species, I would say that effect comes from some normal distribution with a mean and a variance. And that mean would then be what's that average effect of forest cover across all my forest interior bird species. And that standard deviation would be how much variation is there among the species in their effect of occurrence on forest coverage. So here's that example kind of showing that for these biological parameters, right? You'd have all these single species level effects, and then you have some mean effect across the community. So why do you want to do this hierarchical community modeling? Well, there's a couple of reasons. The first is that models reflect how species are distributed and how they assemble to form communities. So if we want to estimate things like species richness, a really nice way of being able to take this approach of doing this is that we're really estimating, okay, which species is here on top of what other species is here, what other species is here, instead of, sort of more common non-parametric approaches where you're doing things like assuming the number of species has to saturate, but it's not based on anything biological. So let me give you an example with some work of ours that we did. So I collaborated with this student, Zabi, who was visiting from Spain, and he was interested in understanding how irrigation, how the implementing irrigation affect bird communities in local areas in Basque Country. So 2008, all this yellow area was areas that were non-irrigated. And then what you can see in this particular landscape over time, by 2019, a lot of the area had been irrigated. And the question is, how does that affect the occurrence of bird species? So we've developed this community model, and we thought about aggregating species into, different groups based on habitat associations. And what we found is that many species, even from farmland, shrubland to non-specialist species, mostly their change in occurrence decreased, right? So they had a decrease in occurrence probability once the irrigation was implemented at a particular site. And of course, though, what's cool about this is we don't have to assume that every bird responds the same way. There were, in fact, of course, some species that increased. And then what's interesting about this is we can aggregate this to look at species richness across time at the sites that were never irrigated. So those are yellow sites. And then once irrigation happened in these blue sites. And what we can see is we're able to use this community model to estimate explicitly how much species richness dropped at these sites and uncertainty metrics around that. So another reason we want to develop these hierarchical community models is that we get increased precision in species level parameter estimates, especially for rare species. So this can be important when we're trying to understand what's going on with the species for which they occur rarely in the landscape, or they're just very hard to detect. They're elusive. So this, on a project that I worked on, looking at what was happening to tropical snakes in locations where amphibians had been disappearing related to the fungal pathogen chytrid. And so this study took place in Panama, and essentially a collaborator of mine was walking these transects, documenting the loss of amphibians related to the BD fungal pathogen, so this chytrid that causes mass mortality of amphibians over about a 12 years. And during this time, my colleague, Karen Lips, hypothesized that, yeah, I really believe that we're seeing less snakes, but snakes are so rare in this area and so elusive that it would be not possible to use those data. She was documenting every snake that she'd seen, but it wasn't possible to do any kind of standard analysis, anything like a standard occupancy model. So what we were able to do is estimate the change in the occurrence probability from before the chytrid had arrived to the location to the time afterwards when when the amphibians had all declined. And you can see on this right side of the graph that there is a very, very low number of detections. And this is out of, hundreds of sampling events before and after. So I'm talking like 600 sampling events, something maybe before and about 600 after or so. And we see very, very low detections of the snake species. And mostly that's just because not that they're not there, they are there, but there may only be one individual of the species within the sampling area. And they're very, very elusive. You could be walking a transect, the snake could be right next to you and you won't detect it. So we weren't able to estimate the occurrence probabilities that precisely, but we were able to look at what was the probability that the occurrence probability had decreased after the arrival of chytrid and the loss of amphibians. And so that was a really interesting process of where we were able to document these declines in snake occurrences after the loss of amphibians. And kind of around that same idea is that we did that for individual species, but we can also use hierarchical models to produce this community effect on other biodiversity metrics. So in this same example, we were able to estimate the probability that richness had declined. So even though only 30 species were detected before the amphibians declined and 21 after, we actually estimated that the number of species was something like 52 and had declined to 40. So we could look at the probability that richness had declined. Here was 85%. And then on the right, we were looking at the composition. So this is non-dimensional PCA type model, multidimensional model, where we're looking at, okay, the sort of niche space that the community takes up. And after the loss of amphibians, we found that the remaining snakes were more similar to each other than they were prior. So we see this change in the composition of the species. In another example where we were looking at how do diverse bird species exist in tropical forests, this was taking place in the Albertine Rift, led by my graduate student, Sam Abar, to try and understand how so many diverse species live in such a small area. And that's a whole nother interesting project that I don't have time to go into, but I'll just show you we were able to look at sort of these community effects and look at, all right, overall, where do these species live relative to the elevation? And we could see like most species, peak around 2,400 meters, something around that, but that there's, different species using different parts of that. So really this nice aspect of the hierarchical community model is that we can understand something individually about the species and also about the community as a whole. Okay, so that's a summary of what are hierarchical community models and why we would use them. And now I want to switch and talk about the same thing for single species integrated models. So let's start with what are single species integrated models? Well, let's talk about the data first. So before we talked about that a single survey or single data collection method or sampling protocol that we're doing might collect a lot of information about multiple species. But on the flip side, there's also a lot of information that one could collect about an individual species. So I'll use an example of birds, for example. We could go out and let's just think of an individual species. We could go to a location and we could record detections, non-detections of it. We could count how many are there. We can do productivity surveys. So we can look at, number of eggs produced or number of eggs or number of individuals that fledge nest. And of course, we can do stuff like capture recapture, where we mark individual species and then follow them through time to see how many survive and come back to certain locations, all that sort of stuff. So this is kind of the flip side where we're thinking about one specific species, but we're collecting a lot of different types of information about that species. And the idea with the single species integrated model is we want to put those multiple data types on that single target species together into a model to learn more about the species than we would be able to do if we just analyzed each data set separately. Okay, so let's talk about the mechanics a little bit. The first step when we're thinking about an integrated single species model is that we want to think about what is this biological process model. And that can be really detailed. So here I have just a really simple two-stage model of NJ, which juveniles, and NA, adults. And I'm just thinking about this here is just a diagram that talks about, all right, we get adults because juveniles survive into adults. Adults can survive and stay adults. Adults produce individuals, right? So that's what that F is. New individuals can immigrate in. That's what that omega is. And so we could develop a model that looks at the biological process. And the kind of model we're going to develop really depends on the kind of data we have. If we have detailed data on survival, or fecundity, we might be able to develop this kind of real, classic population model, like a Leslie matrix model or some other thing. However, that's not always the case. Sometimes our single species models, our biological process is more phenological, where we're just seeing what happens. So in this case, like this is another example. Let's say we didn't have that kind of detailed capture-recapture data. Instead, all we had was maybe counts or estimates of occurrence at a particular spatial location. Here, I've got this map of Michigan. You can see this dot on there. And let's say instead, what I want to do is relate the occurrence probability of the cerulean warbler in Michigan relative to the forest and vegetation cover. So there, maybe I'm doing something simple and I'm just, doing some sort of linear model where I relate these covariates to the occurrence probability. So we specify this biological model, and the way we think about it kind of depends on our question and maybe the data that we have available. So then what we need to do is we need to link those data that we had, those different data sources that we had, to our biological process model using what we call the observational model. So here is what I've got: multiple different data types represented by these blue diamonds. And there's productivity data, maybe. So that's like number of individuals that are produced per adults. Let's say Y are our count data. M might be our capture-recapture M array, and maybe there's some other data available too. And what we want to do is see how each of these different data sources links to those parameters, those biological process parameters that we had just previously defined in the next step. So for example, our M array, our mark recapture data, that gives us direct information about juvenile survival and adult survival, but we actually also get that information from the count data, from accounting just how many there are through time. And so we can put this together in a model where we link those data that we have to the specific parameters in our biological process model. And of course, what we have in all of these is always nuisance parameters, because again, what we talked about previously with the community models, we don't observe things perfectly. So there's a sampling process that we have to account for in our analysis if we want to get unbiased estimates of things like survival, reproduction, immigration, that sort of thing. Okay, so that's the basic idea for an integrated model. But now let's talk about why would we want to do this? Why would we want to do a single species integrated model? So the first is that it increases accurate precision in biological estimates. So let me give you an example here. This is taken from work that my graduate student, Matt Farr, conducted. This is a simulation, let's say, where on the left, we've got an ecological predictor. You could think of it again as forest cover. And those values from this orange to green are just values of this ecological predictor, standardized on some scale. And what we want to know is kind of the abundance overall and the effect of covariates. So here we're going to say this overall abundance, which is like our intercept, our average abundance, is this blue line. So that's truth. Same thing with the effect of the covariate. It occurs at this value 1, let's say, and we want to be able to measure it. Well, the first kind of data that we might have, the lowest, cheapest, easiest data to get, are what we call presence-only data. So that's just when someone's gone out and only recorded when it's there. This might be something like you could think of like iNaturalist. Generally, scientists don't collect data this way, but there's a lot of volunteer participatory science programs that do. So if you go out on the landscape and you just record where the species is, you can get an estimate of the covariate effect, so how this covariate affects the occurrence of this species, relatively imprecisely, but you cannot get an estimate of what the overall abundance is. And that's because in this model, with only presence-only information, it's just unidentifiable. It's not estimable. However, if we add another structured type of data, in this case, we're thinking about distance sampling data, we can quickly get both an accurate number of that intercept on the total abundance, and we get increased precision on our estimate of the covariate effect. And of course, even a tiny bit, so if we only 5% coverage on the landscape, we get markedly improved estimates, but you can see that the parameters get even more precise as we increase the amount of data up to 50% coverage on the landscape. So that's just one of the first things. If we put together different kinds of data, we can get more accurate, more precise biological estimates. One other thing is this single species integrated model can help us understand how processes change through space and time. So one of the things I work on, and I alluded to at the beginning, was this idea of macro systems biology, which is the idea that what's happening, what we're seeing across space, especially large space, is a product of both what's happening at a local scale, but also what might be happening at a really broad scale. For example, climate as opposed to weather, that sort of thing. So when we think about the types of data that we have, let's say on an individual organism, at a broad scale, we tend to have unstructured data with lower information content. So think back to what I was talking about, like iNaturalist observations. We might be lucky to have volunteer scientists that are going out and recording an individual or something, and they might be doing that across a really broad space. We might have that for the whole continental US. But this high information content data, structured data, right? That tends to be spatially and temporally limited. And we can think about that, just thinking about something like capture data. It's difficult in most cases to get that kind of really, really detailed information across a wide space. And also usually across a long time frames, right? It just takes a lot of effort. So there's a benefit in putting together these different data types because they can let us understand something really detailed at a local scale, but also understand what might be going on very broadly. So I'm going to give you an example here with black terns about how single species integrated models can help us understand the factors that are influencing population dynamics. So black terns have been declining. This data, again, is from locations in Michigan. They're very hard to sample and they occur pretty low abundance. So this is a colony where data have been collected since 2013, and we can see the counts are declining and so are the estimates. And what we did, because there's multiple different data types, there's productivity data, there's capture data, is we put this all together to estimate, okay, can we understand what's going on with the juvenile survival, the adult survival, and something about the fecundity. And using this approach, this integrated community model, this integrated single species model, we were able to estimate these parameters for black terns that really just don't exist elsewhere. And what was interesting is we used them to understand, all right, which of the specific factors or of the demographic parameters are likely causing declines, and which, if we change, could change our population dynamics most dramatically. So this here is a quasi-extinction probability with different management scenarios plotted under different NAO scenarios, which aren't important. You can focus on just the solid lines here. But essentially, we can get the most bang for our buck by increasing adult survival. And that, I'm just going to go back one slide. That may not be so obvious because the adult survival is pretty high, but there are variabilities from year to year. And juvenile survival is fairly low, which is something we know when they fledge, the likelihood that they're coming back is pretty small the following year. But if we could get some more stability and increase that adult survival just a bit, that would have the biggest effect on helping to maintain this population of terns. Okay, and then I'm going to talk a little bit about monarch butterflies because this is a project we've worked on for a long time where data integration has been really helpful. And a group that I've been working with at the USGS Powell Center, we just published a paper that came out on Thursday in Science about butterfly declines across the continental US. I'm happy to answer any questions, but I'm not going to go too deep in that. We did use some of the methods that I'm talking about here, but basically in that analysis, we showed that there had been a 22% decline of butterflies from the year 2000 to the year 2020, so a loss of 1 in 5 butterflies. But I'm going to talk now for a second about monarch butterflies here. So monarchs in particular, they have this really broad spatial extent in Eastern North America where they overwinter in Mexico. They spend the spring breeding in Texas and the surrounding areas. They move up everywhere east of the Rocky Mountains and produce 2 to 3 more generations. That final generation at the end of the summer migrates primarily from the Midwest area, upper Midwest area, migrates all the way back down to the overwintering site. So they've got this really unique life history. They're iconic and they're really widespread. And they've been declining. So the question is, why had monarchs been declining? We developed this integrated model, put them together, all the factors that have been affecting monarchs across their full annual time series and full seasonal effects, and determined that spring and summer weather conditions were more than 7 times more important than other factors in explaining variation. So there had been different hypotheses about, was it changes in the winter population? Has it been crops and herbicides? Well, at least since the last couple of decades, so about 2004, we've seen that weather and climate patterns have been mostly driving the changes in monarch population abundance. So following along this same trend, what's really great about these integrated models is we can use them to predict the trend and future vulnerabilities. So with that same monarch thread, what we did is we predicted how big the population sizes are likely to be in Mexico in the forest. And this top level is showing, okay, how big the population might be in the near future, the mid-century, and the end of the century under various climate change scenarios. And then the bottom shows the number of years, so how risky it is that the population dips below its ever historical minimum, which was measured as 0.67 hectares, right? So what you can see here is in the short term, we don't find that much difference by the difference climate change scenario, but longer term, towards the end of the century, we're going to get really different results based on whether or not we have business as usual, so our high climate change scenarios, or whether or not we're able to curb in some of the fossil fuels that we're putting in and mitigate climate change. And so what's cool with this is we can use this information to predict, okay, future trends and vulnerabilities and where might be good climate refugia. So these are on the left expected counts of monarchs and counties across the Midwest breeding season. So you can see this like kind of upper Midwest has the highest. This is over from the historical time period since 2004. Upper Midwest sees the most monarchs. But what's interesting is in the near future, in the next 20 years or so, areas like Ohio, northern Ohio, southern Michigan, eastern Indiana might end up being really good for monarchs because we could see an increase in their abundance there. And that's because those areas are projected to not get too much warmer and stay pretty wet. So that could be a good potential refuge for monarchs, at least over the next several decades. However, once we start to see really high emissions, later in the century, it looks like most of this map gets red, which suggests a decrease in monarch populations. So, these kinds of models can be used to look at, all right, what's the vulnerability and maybe where spatially are things looking better than other places. Okay, so we talked about the hierarchical community model, and then we talked about the single species integrated model. So now we want to put that together into the integrated community model. So what are integrated community models? Well, many surveys produce multiple types of data on multiple sympatric species. So the first one with the community models, we talked about a single survey that produced data on multiple species. And then in integrated models, we talked about the fact that for an individual species, there are many different types of data that one might be collecting or many different surveys that they do. But the reality is we have both of those scenarios for lots of species. So that means that many species, we might have multiple different types of data, or even the same type of data, but collected in multiple different ways using different protocols. So it might be count data, but the counts could be collected in a different way, different time, different methodology. All right, so the idea with that is we want to integrate all those multiple different types of data on multiple different species to analyze what's going on simultaneously. So what are these integrated community models? So the first is the basic idea, what we talked before, is that we want to understand an individual species latent, let's just make it simple, occurrence probability. We want to understand what is this, does this species occur or not in a particular location and at a particular time? And what we think affects that is environmental covariance. Now, what we have is multiple different data sources on that individual species, right? So we're going to say for that species I, we might have one data source that's a presence only. We might have another one where it has repeated counts, all sorts of different kinds of data. And so now what we want to do is put those together in that kind of single species integrated modeling framework. And then we want to link each of those individual species parameters at the community level. And that gives us information both about the individual species and gives us something to understand about the community as a whole. So why would we want to do this? Well, this combines the advantages of hierarchical community models with single species integrated models. We get species level parameters. We incorporate multiple different types of data and sources. We estimate parameters, sometimes even without explicit data. That's another thing that we can do. We can look at biodiversity metrics, and we can gain inferences on data deficient species. So there's a lot of potential advantages to that. And I'm just going to show you a few quick examples of the why from some of the work that we've done. So here is an analysis. This is simulated data on a paper published by a postdoc of mine, Neil Gilbert, who's now a faculty member at Oklahoma State. And he was looking at, okay, what happens if we can buy single count data, so like something where we walked a transect and did some counts, with distance sampling data? How well do we estimate parameters for individual species? So here we've got two types of species, the common species and the rare species. And what we might find is for the common species, that have a lot of data, maybe we don't need quite as much integrated analysis. But for the rare species, so those are the like dark blue species, we find that we get better estimates generally when we use multiple different data sources. And that in most cases, the community integrated model, so that's that highlighted one on the top panel, and then the bottom panel in bold, that gives us the best estimates and most accurate and precise estimates. So accurate would be centered around 0, precise would be that the boxes are thin and the arrows are not that wide under the community integrated model. So we can understand them both using this approach, combining the data. We get the best estimates for abundance, the effects of covariates on abundance, how our detection influences our observations, and anything related to covariates on that. So, and then what Neil also did, which I thought was pretty interesting, is he said, well, what happens if not all the assumptions are met? So what happens, for example, again, taking the same thread where we have different amounts of data for count, this like lower, this lower quality count data, and this detection, non-detection data. So, or sorry, lower quality count data, and then our higher quality distance sampling data. So in this top panels where it says same detectability, we find that even if we have this lots more of the lower quality data, by combining everything together, we still can get the best estimates. But what happens if some of that lower quality, lower information content data is a little bit modeled incorrectly? So in this case, we assumed, for example, that our detection probability was 20% lower than in our distance sampling data, but we didn't have anything to estimate that. We find that in most cases, particularly for the covariate effect, that doesn't really bias our estimates. We might get some bias though, however, in the absolute value of those counts if we misspecify the model. But generally, the idea here is that it actually works pretty well, especially for estimating that covariate effects. So why else? So here's an example where we integrated bird data to help us understand how species within communities vary across their spatial trends. And in this case study, this was work done by a postdoc of mine, Jeff Dozer, who's now a faculty member at North Carolina State University. We integrated NEON data with other data, BBS data, National Park Service data, that sort of thing. And we estimated these trends. And what we find is that not all the different data sources give us all the same trends. And that could be a couple things. I mean, it could be biases in the data, but it could also be the difference of the placement of those. For example, the BBS are roadside surveys where NEON data are occurring in, more pristine habitats. So these might be reflective of really what's going on. Like, for example, we might be seeing really different trends of the black-throated green warbler in roadsides compared to in protected areas. So this approach allows us to look at that explicitly and calculate our uncertainty on it. We can evaluate broad-scale spatial trends by doing this. So same approach here, where we're integrating these different data types, including NEON, eBird, BBS, and national park data. We can understand what's going on for both a common species, such as the black-throated green warbler, which has an occurrence probability across very many states in the northeastern US. Whereas the cerulean warbler, which just doesn't have as much data, and without doing this combined approach, we probably wouldn't know what's going on. But what's also really cool about this is that top left panel, this integrated community modeling allows us to estimate with uncertainty what that species richness map looks like across space. And I think that's pretty cool. I mean, of course, we could do each species separate, each one by one species, and then aggregate this together. But when we put it in this framework, combined framework, we're able to account for all of that uncertainty. And then the final is this effect of forest cover. So that's panel B. And that's just showing what's going on for this effect for every species. So here they're labeled by how large that effect is, where essentially what we see is all of these forest interior species have a positive effect of forest cover, and then we can see that mean value, which is above the black horizontal line. So that's a mean across the full community. And then here's an example with butterflies. In this case, this is just butterflies in the Midwest, where we were looking at the trends through time and trying to understand, okay, what's the probability that each of these different species are declining across the whole of Midwest by integrating data from multiple different states and the North American Butterfly Association citizen science data. And then finally, we've got this project kind of in the works where we're doing this full on, which we call MIPM, which is the multi-species integrated population model. Now, this actually does happen. There are certain cases where we do have multiple survival capture data for multiple different species. And we're applying this to a community of Arctic shorebirds. So Kayla Davis is leading that work. She was a graduate student in my lab, and now she's a postdoc at University of Saskatchewan. And so we're putting together these integrated multi-species integrated population models where we've got the full annual cycle where we're modeling survival, modeling recruitment, and linking those at the community level. Okay, so I just want to talk a little bit about next steps for what we're thinking here as the data grow and we're thinking about and we're learning more about our processes. One is that we're really extending to macro systems. So I have a grant from NSF from the Macro Systems Biology, now defunct Macro Systems Biology program. But essentially what we're trying to do is look at what have been the factors, land use versus climate change that have led to those 3 billion birds lost across the continental US. And here, I think this is one of the cases where the different data sources that are shown up in the left corner. So this is NEON, BBS, this is national parks across the country, and eBird data. Each are surveying different types of landscapes, right? So NEON, again, is these protected forest interior sites, same with NPS. Whereas BBS is roadsides and eBirds are where people bird, which, I mean, we do all know that that's happening over a really large area. But again, they're going to the best birding areas. So we really are able to, include more by looking, for example, at roadsides, at using that with the BBS. So we're putting together this model to look at the effects of land use and land cover changes through time and climate to understand how much of individual species and biogeographic communities as a whole have changed related to these variables. So that's, I kind of jumped the gun here. It's a little bit of a schematic showing this, which is just not really important, but trying to think about like the math through how we account for all of the spatial correlation, the spatial variations to look at those factors behind continental scale bird declines. Another thing that we're doing too is we've started looking at regional butterfly trends. So I alluded to those national ones, and Wendy Leuenberger, who is a graduate student lab, actually just submitted this paper to PNAS where we're looking at, okay, what are the trends of butterflies across a longer scale of time? And are those varying by traits or by different related groups? And ultimately, what are the kinds of drivers that are causing those? Can we tell how much the climate and weather variables are leading to observed changes in butterfly communities? So that kind of wraps up sort of where we are, but one of the things I really want to talk about, which I think is important, is the idea of developing a generalizable framework for this. So, I started off by saying, like, we're entering a biodiversity crisis. We're seeing a lot of species lost, and we're in this position where we can no longer be, assessing species by species and developing conservation plans for species individually. So I think we really need to be thinking about what is the general modeling framework so that we can get, very quickly an understanding of what's going on with species that can get translated into action, to figuring out what we can do to protect all these species simultaneously. Yes, so that's pretty much it. I'd like to thank the people in my lab, and particularly here's the lab and other collaborators and funders who have made this work possible. And I'm happy to take any questions in the last few minutes here. Thank you.
All right. Thank you so much, Elise. That was great.
I don't know if you want me to stop sharing, or what do you think?
I think it's fine to share if you want to go back to any slides.
We can go this one.
Cool. All right. So Samantha mentioned in the chat, please add questions to the Q&A or add questions in the chat. We have a couple in the Q&A. So the first one is from Fabiola Rodriguez. And the question is, can you integrate data sets that do not match perfectly in spatial sampling? So fewer
Yes.
sites for capture, recapture, than counts, et cetera. How about temporal sampling mismatches?
Yes. So this is a great question. And we wrote a paper trying to talk about, it's a challenge. Don't get me wrong. It's not like, oh, we can just do this, but there's lots of approaches that people are developing for those mismatches. And that's key, because the reality is all the data are mismatched, right? If they weren't collected thinking that we were going to analyze them together. So yeah, there's a lot of different strategies out there, and it depends on the specifics of, how the mismatch is, but we can add covariates, we can add random effects, we can add structure into those kinds of random effects and account for those variations. But yeah, the answer is yes.
I have kind of a follow-up question to that. And so it seems like there's a lot of folks that, want to apply AI and ML techniques to these sorts of questions. And so I'm wondering if you used any of those sorts of tools and sort of this multimodal data integration. And I'd be curious to hear your thoughts about that, the advantages, pitfalls, things like that.
Yeah, so I think there's a couple of things. So first, I think there's a real opportunity for AI in terms of processing a lot of the data that we have available. So if you think about now, I'm sure everybody at Neon's familiar too, right? There's a lot of data in there, processing data. You've got these big data, in some cases they might be like heavily autocorrelated, they're not independent, and thinking about ways to process them in a reasonable way could be something that I think AI can do. Another example, right, is like these automated camera trapping data, where we're trying to sort out, for example, what species are we seeing? Can we tell if it's the same individual? All that sort of stuff. So I think there's a lot of potential opportunity with AI to get at some of those questions. So I think number one, what I call harmonizing data, where we're putting data together either by identifying or matching, I think is a huge, huge possibility. So the other question about the AI or, other kinds of non-parametric models, I'll say, is, so there's clearly value in that. I used to think there was no value really in like non-parametric approaches. I couldn't really understand it, because I'd be thinking, well, all right, sure, I can like describe what's going on, but I don't know why it's going on. But I've been convinced that in some cases, like understanding just what was there and not why things are there can be really helpful. We see people do this kind of stuff, for example, like with trees across space. Like, we don't really know, what are the factors that are leading these mixed species, stands or whatever, but I can describe them very well with things like spatial parameters or non-parametric approaches, which would, I think, include AI. So I think there probably are benefits for that, but a lot of what I work on is really this question of why. Like, why are we seeing what we're seeing? If you think about like, what can we do about it? The what can we do about it really necessitates a deep understanding of why. And I'm not convinced that AI is going to be that helpful with that, right? Because just because you can find patterns it's kind of like a model. Like if I have 300 data points and I make a model with 300 parameters, I can fit that perfectly. But then I want to be able to predict to the next thing, I'm almost certainly not going to be able to predict to that next point, right? So, I mean, you saw some of what I was showing with like prediction. All right, I want to know where's going to be maybe good in the future. And so I think it's helpful to think about the relationships between variables and trying to understand that in parametric ways. But I'm open to being convinced. There could be, new approaches out there with AI that somehow can predict the future. But yeah, we'll see. I mean, technology is getting fast, and our computational systems are getting even faster. And I think that presents an exciting opportunity.
All right, Samantha, how are we doing on time? A couple more questions.
Yeah, we can go. Just leave me 1 minute at the end for wrap up. But yeah, take another one or two for sure.
Okay, I'll go to this one here. So from Ellen Hurlbert, great talk, Elise. How would you describe the minimum data requirements for these integrated community models on a per data source? So
Yeah.
for example, just 100 unstructured observations with 20 structured survey. Does it become worth considering? When does it become worth considering this approach?
I think that's a really, really good question. There's no simple answer for that because it's going to depend on your question, your data type, and your model structure. Remember at the beginning, we talked about like, oh, your detailed biological model versus, like a phenological model. Oh, I'm just relating this covariate, the occurrence of this species to, some environmental covariate. So there's not a great answer for that, unfortunately, but there are ways that you can diagnose it, right? So I would always say if you're thinking about the integrated approach, you're definitely going to want to do preliminary analysis where you're analyzing things separately, right? So for all this stuff that we're doing, we're taking our individual data, maybe not necessarily by always individual species, although sometimes, but we're definitely analyzing those data separately to understand what the limitations are, like what we don't know about it and how much we can trust it, right? And I think your point about structured versus unstructured data is really a good one because you can have 20 unstructured data points, but they're maybe not the same usefulness as one data point that were collected in a structured survey design. So I'll say it depends. We've had some papers written about this and other groups have written about this too, but more ways that you could target figuring out this specific problem for your individual data set and questions.
Right. And then we have a couple questions that I think are generally similar. So one is, can you talk about the differences, approaches to integrated community modeling versus hierarchical community modeling in a little more detail? And then the other one was, can you talk a bit about the main assumptions behind the integrated community model, and do they differ between the different types of models?
I'm trying to pull up the things because it's because I'm thinking I didn't totally get the first question. Okay, so assumptions. Let me say assumptions are going to be all the assumptions of the individual models, right? So if you're doing a hierarchical or if you're doing an integrated community model, you're going to have to do all the assumptions of the hierarchical model and all the assumptions of the single species model. There might be other ones on top of that, but that will depend on the exact structure of your model. I think the question was, what's the difference between hierarchical community and integrated community? Was that, and so the hierarchical community is just a community model with only one data source, and the integrated community uses multiple data sources. So that's the difference for that. Whereas the single species integrated model uses multiple sources for a single species, and the hierarchical community, right, and the integrated community model uses multiple data sources on multiple species. I should make a diagram. That might be helpful. But yeah, the assumptions is a good question. And you're definitely going to want to check that as you use these because, and there's ways, again, that you can diagnose it to make sure like, okay, am I, missing this assumption and it's leading me to something crazy? Or am I missing this assumption and it's not really that big of a deal? But there are ways that you can kind of diagnose that.
Cool. So I think we're at the hour now. Real quick before Samantha has her minute to wrap up, I was wondering if you have any like resources that you'd want to point people toward, like a
For
tutorial
this
or a paper or anything
models,
like that
for these
for your
models,
approach. Yeah.
You know what? While Samantha's talking, I can put in a link to our integrated community model paper where we introduced the idea. So, I mean, there's other papers too, and a good resource would be to look at who cited our paper, because I think there's people doing more exciting stuff about that. But I'll put the link into our paper while Samantha's talking.
Cool. Well,
Wonderful.
thanks so much. I'll give you
Yeah,
a
thank
full
you.
applause.
Thank you. I'm going to stop sharing now. Thank you.
Yeah, that's great. So, wow, what an interesting and thought-provoking seminar. We're so grateful to have had Elise to present to us today. We heard a little bit about integrating NEON data there at the end, so we're excited to see what more comes out of that. This is a monthly science seminar series, so next month we're going to be hearing about plant community spatiotemporal trends in a Hawaiian forest, where we have our one and only Hawaii NEON site. So if you're interested in tropical plants, please come and join us next month. We'll give Elise one more minute here to drop the foundational seminal paper on her modeling approach into the chat. I think I see it. So if you're still on the call, go ahead and grab that link. Thanks again for a really wonderful presentation. Thank you all for attending. We look forward to seeing you at another seminar soon. Thanks so much, everyone.